Good afternoon. I'm Shelly, a graduate student at Professor Isaac Quitz's lab, and I'll be talking today about macro and micrometastasis in neuroblastoma and their interactions with the microenvironment. Neuroblastoma is the most common solid tumor in children. It originates in the sympathetic nervous system, most commonly arising in the adrenal gland. The preferential metastatic sites are lymph nodes, bone marrow, liver, and skin. Lung metastases occur when tumor is widely disseminated. It is now widely known that the interactions between the microenvironment and the cancer cell have pivotal role in determining the fate of these cancer cells. There are few possible fates for cancer cells: progression towards metastasis, apoptosis, or entrance to a dormant state. There are two types of dormancy. The first one is cellular dormancy, which is a G0, G1 arrest, in which cancer cells are truly inactive. The second type is tumor mass dormancy, which is valence, proliferation, and apoptosis, in which cancer cells aren't truly inactive, but are incapable of expanding. Cancer cells that enter these dormant states and that are clinically undetectable are termed micrometastasis. Since the interactions between the microenvironment and the cancer cells have pivotal role in cancer progression, our main objective was to identify microenvironmental factors that are associated with neuroblastoma progression. The experiments that I present to you today focus on the role of the microenvironment in determining the fate of micrometastasis. For these and other objectives, we developed an orthotopic mouse model for human neuroblastoma metastasis. We inoculated the human neuroblastoma cell lines, MHH-NB11 and SHSY5Y, to the adrenal gland of nude mice. The adrenal gland is the orthotopic site for this tumor. That we yielded the adrenal cell variant, which will be called the local variant, and after 4 selection cycles, the lung metastatic variant, which will be called macro-lung metastasis. Immunohistochemical examination using synaptophysin, a neuroblastoma cell marker, did not detect metastasis in the bone marrows of these mice. This was rather surprising since there is evidence for minimal residual disease in the bone marrows of neuroblastoma patients. Another interesting result was that none of the mice that were inoculated with the local variant had detectable lung metastasis, while half the mice that were inoculated